A caterpillar climbs toward the light. Not because it chooses to. A virus has rewritten its final hours. Elsewhere, influenza steals pieces of the cell's own messages. HIV does something quieter. It inserts a DNA copy of itself into the cell's genome. And a bacteriophage turns a bacterium into an assembly line. None of them has a brain, no muscles, no intention. Yet each redirects a living system from within, and each uses a different kind of biological theft. Some seize behavior, some steal molecular tools, some hide inside the genetic archive. The strangest does not merely invade a cell, it builds a factory inside it. 6 viruses, 6 completely different hijackings. And the first begins inside a caterpillar. Baculoviruses are insect specialists, not human pathogens. Their most dramatic work happens inside insect larvae. Infection often begins when a caterpillar eats contaminated leaves. Protective protein crystals enter its alkaline gut. There, the crystals dissolve, releasing virions across the gut lining. The infection then spreads through the caterpillar's tissues. Soon, its normal schedule can begin to change. Some infected caterpillars become unusually active before death. In certain host virus pairings, they also move toward brighter areas. That movement can carry them higher through the vegetation. From there, viral material can disperse across more leaves. But no single switch explains every infected caterpillar. Viral genes such as EGD and PTP can contribute, depending on the virus and host. So this is not literal mind control. It is infection-altering physiology, movement, and environmental responses. The result still looks almost impossible. A creature climbs and dies high, and may help the infection travel farther. But the next virus does not pull upward. It follows nerves into darkness. Rabies virus is shaped roughly like a microscopic bullet. Its outer envelope carries surface glycoproteins. After an infected bite, the virus near the wound can enter peripheral nerves. Then it travels inward using transport systems inside neuron. The path leads toward the spinal cord and brain. That journey can take weeks or months. During this quiet interval, a person may feel completely well. Once central nervous system disease appears, the situation changes brutally. Furious rabies can produce agitation, confusion, and difficulty swallowing. Paralytic rabies follows another pattern. It's dominated by weakness and paralysis. The virus can also reach salivary tissue. That connects neurological disease with transmission through saliva and bites. It is tempting to call this behavioral control. But science must be more careful. Rabies disrupts brain function. It does not issue precise commands. The apparent strategy emerges from disease, anatomy, and opportunity. And one fact matters beyond every cinematic image. Rabies is preventable when exposure is treated before symptoms. If exposure may have occurred, urgent medical care matters. Rabies changes an entire nervous system. Influenza steals something much smaller. Influenza A usually arrives inside an enveloped, spleomorphic particle. Some particles are round, others stretch into long filaments. Their surfaces carry hemagglutinin and neuraminidase proteins. After entry, the virus releases 8 genetic RNA segments. Those segments travel into the cell nucleus. That is unusual for an RNA virus. Inside the nucleus, influenza faces a basic problem. Cellular ribosomes expect messenger RNA with a capped beginning. Viral messages need that cap too. But influenza does not build one from scratch. Its polymerase grabs newly made host RNA. One polymerase subunit recognizes the cap. Another cuts away a short capped fragment. The virus uses that stolen piece as a primer. Scientists call this cap snatching. The phrase sounds like metaphor. It describes a real molecular reaction. A host message becomes the starting handle for a viral message. That hijacked message can then reach cellular ribosomes. Influenza has stolen legitimacy, not just material. To the ribosome, the capped viral RNA now looks usable. One tiny modification opens the cell's production system, yet influenza remains outside the permanent genetic archive. The next virus crosses that boundary. HIV-1 approaches a CD4 cell inside a host-derived envelope. Beneath that envelope sits its famous cone-shaped capsid. Its binding begins with CD4 and a core receptor. The viral and cellular membranes fuse. The core enters, carrying RNA and essential viral enzymes. Reverse transcriptase copies the viral RNA into DNA. That changes the language of the infection. RNA has become a DNA version. The viral DNA reaches the nucleus. There, integrase helps join it to the cell's DNA. This integrated form is called a provirus. It can remain quiet, or become transcriptionally active. When active, the cell helps produce viral RNA and proteins. New particles assemble near the membrane and bud outward. Viral protease helps mature them into infectious particles. Integration is why simple removal is so difficult. The viral sequence is no longer merely beside the cell. It has joined genetic material the cell can copy. Modern antiretroviral therapy can block multiple stages of this cycle. That allows people with HIV to live long, healthy lives. But integrated viral DNA can persist in cellular reservoirs. That is not a violent takeover. It is molecular concealment. HIV hides inside the genome of a nucleated human cell. T4 chooses a host thousands of times smaller than us. A T4 bacteriophage cannot infect a human cell. Its target is certain strains of Escherichia coli. Its body looks almost engineered. An elongated icosahedral head stores double-stranded DNA. Below hangs a contractile tail, a base plate, and long tail fibers. The fibers encounter bacteria through random motion. Molecular fit determines whether attachment holds. Once attached, the tail sheath contracts. The viral genome passes into the bacterium. Most of the outer phage structure stays outside. Inside, T4 begins a precisely timed takeover. Early viral proteins redirect the host's transcription machinery. Host DNA is broken down as infection advances. Some components can be recycled into phage DNA. Viral genes organize genome replication, head construction, tails, and fibers. These structures assemble through partly independent pathways. Empty heads are packed with DNA. Tails and fibers attach afterward. The bacterium fills with new phages. Finally, viral proteins rupture the cell envelope. The bacterium opens and releases assembled particles. It really has become an assembly line. Yet T4's factory is distributed across one tiny bacterium. The final virus builds something much more visible. Mimivirus was first mistaken for a bacterium. That mistake makes sense. Its particles are enormous by viral standards. A dense layer of fibers covers its pseudo-icosahedral capsid. One special vertex forms a five-armed stargate. Its known hosts include free-living amoebae. An amoeba can engulf the particle as though it were food. Inside, the stargate opens. The viral core is released toward the cytoplasm. Then the infection reorganizes a region of the cell. A distinct viral factory appears in the cytoplasm. This is not a metal building. It is a concentrated biological zone. Its viral DNA is copied there. Viral genes are transcribed there. New capsids begin forming around the factory's edges. Genomes are packed into those growing shells. Its fibers are added as the particles mature. Soon, one cell contains a crowded viral production center. The virus has not borrowed just one tool. It has rearranged an entire cellular neighborhood. That makes Mimivirus a fitting final chapter. It blurs the line between a simple particle and a complex biological system. It's not alive like a cell, not inert like dust, something dependent. It's elaborate and still difficult to define. A caterpillar climbs toward the light. Now that image belongs to a larger pattern. One virus shifts behavior. Another travels through nerves. Another steals caps from messages. One hides in DNA. One converts a bacterium into a production line. And one builds a factory inside an amoeba. None thinks. None plans. Yet evolution has shaped precise ways to exploit living systems. The smallest agents can redirect the largest machinery, not through intention, through chemistry, selection, and time. And once you see the theft, the virus looks different. It's not as a tiny monster, as a set of instructions. It's waiting for the right cell.